our next speaker, Nick Till. Nick is a NIH postdoctoral fellow in Professor Carolyn Bertozzi's lab at Stanford, where he currently employs chemical approaches to manipulate the cell surface through various induced proximity modalities. Nick earned his BA from chemistry from Reed College, where he conducted research on bismuth catalysis in Professor Rebecca Lalonde's lab. And following internships at Gilead and Genentech, Nick went on to pursue his PhD in chemistry at Princeton under the guidance of Professor David McMillan. And there he developed innovative nickel photoredox dual catalytic reactions and elucidated key mechanistic steps relevant to cross-coupling reactions, which are now widely utilized in both academic and industry settings. Thank you, Nick, for agreeing to present today, and please take it away.
Awesome. Thank you for having me, and thank you for that kind and thorough introduction, Hubert. I'm going to share my screen now. Okay. If everyone can see my screen, I'm going to get started. Like Larry, I'm not going to be talking about targeted protein degradation today, but instead talking about a technology in the induced proximity space that accomplishes the opposite, which is increasing protein levels rather than decreasing them using these molecules called trogotacs. And before I get into the development of these molecules and how we went about this, I want to take a step back and give you some insight into why we were interested in developing this technology. That motivation really starts with the important role that cell surface proteins play in normal physiology as well as in disease. That's true of examples like ion channels, cell surface receptors, and immunoinhibitory ligands. All of these molecules and many other classes of molecules that might come to mind play important roles in the healthy function of a cell and a living system, in functions like ion conductance, in signaling function, and immune tolerance, for example. But when any of these functions go awry, disease can result. That's true in ion conductance, where dysfunctional chloride ion conductance can lead to cystic fibrosis. It's true of overactive cytokine signaling, which can drive autoimmunity. It's also true of dysregulated immunoinhibitory ligand expression, which can drive the progression of cancer. And so if we had a way to intervene and directly manipulate the cell surface proteome, this would be the first step in thinking about how we could address many different types of therapeutic challenges by directly manipulating cell surface proteins. Today, though, the toolkit we have to directly manipulate cell surface protein levels, well, most of these tools lie in the area of loss of function approaches. In particular, targeted protein degradation kind of takes center stage here. It's an approach to removing proteins from the surface of the cell where those proteins drive disease through their presence. Either overexpression or overactivation drives disease, and so removing that protein is a way to address a therapeutic challenge. Some proteins on the surface of the cell, at least in the context of oncology, that might first come to mind are molecules like PD-L1, EGFR, and HER2. All of these proteins, through overexpression or overactivation at the surface of the cell, drive disease. And so this really has been the motivation behind the development of a number of different modalities for targeted protein degradation at the cell surface. Molecules like GlyTacs, KindTacs, AbTacs, and ProTabs are some of the ones on this list, but this list continues to grow to this day and is much longer than this slide allows. We were interested, though, in an opposite challenge. What about those diseases and disorders that are rooted in the absence, deficiency, or dysfunction of a cell surface protein? How then, in this context, could you add a protein to the surface of the cell rather than deplete it? If we had a strategy to address this kind of transformation, this would be the first step to thinking about resolving protein dysfunction, as well as restoring therapeutic targeting in contexts like cancer. In order to accomplish this overall goal, we turned to nature for inspiration, and in particular, this relatively niche biological process called trogocytosis. It's a process by which a donor cell and acceptor cell in a contact-dependent manner can undergo protein transfer from donor to acceptor. When donor and acceptor cell come together, if they undergo trogocytosis, the result is a situation where donor cell and acceptor cell part ways. The acceptor cell ends up expressing protein on the surface of the cell that was originally transcribed and translated in the donor cell, which means this fundamentally breaks the link between gene and protein. Acceptor cell can express protein that it doesn't even have the genetic material to translate and transcribe. Our idea was to co-opt this process using a fairly simple conceptual approach, and I'll outline that here. We thought that with a suitable bifunctional molecule, we might be able to induce contact between donor cell and acceptor cell. Subsequently, we could induce protein transfer from donor to acceptor. And if we were successful in this endeavor, this would be the basis of trogotacs, trogocytosis targeting chimeras, which are these bispecific molecules for targeted contact-dependent transfer of proteins. So what does a trogotac look like? The first proof of concept molecules that we generated look something like this: small molecule antibody conjugates, where the small molecule arm is a CD22 binding small molecule, CD22 ligand. On the antibody side, the first molecules we generated were based on iratumumab, a CD30 binding monoclonal antibody. CD22 has expression that's restricted to B cells. It's a sialic acid binding lectin with well-known endocytic function. This endocytic function is thought to be important for driving trogocytosis. And importantly, for the construction of these trogotac molecules, it has known small molecule binders. It has a natural ligand, which is this trisaccharide shown here, a sialylated N-acetylactosamine, but its affinity for its natural ligand is relatively poor in the double-digit micromolar range. It's not until you move to a synthetic form of this sialicide, this biphenyl carboxyl appended sialicide that was developed by Jim Paulson's group, that now you can get into the sub-micromolar range for binding affinity with the monomer. Throughout the rest of this talk, I'll be using symbols to represent glycans, as these are sometimes somewhat cumbersome to keep track of. The important aspect for this talk, though, is that anytime you see that sort of complicated glycan structure on the bottom right of this slide, that refers to the synthetic sialicide that binds CD22. It's this high affinity CD22 ligand. So that covers the left-hand side of the molecule. What about the right-hand side, this antibody binding arm? This antibody, iratumumab, binds to CD30. CD30 has very interesting biology in its own right, but for the purposes of our proof of concept studies and the rest of what I'll talk about here, CD30 is simply the target of iratumumab. The way we put these trogotac molecules together is actually quite straightforward. We do an NHS functionalization of solvent-exposed lysines on the antibody to install an alkyne moiety. This alkyne conjugated antibody that results can then be further elaborated through a copper-catalyzed azide alkyne click reaction to give the ligand-decorated antibody on the far right. And we do this using an azide functionalized form of the synthetic sialicide I showed previously that we can prepare in 13 chemical steps. We can measure the degree of labeling, or the stoichiometric ratio of ligand to antibody, using MALDI-based mass spec. And under these standard conditions, we get about 6 to 8 CD22 ligands per antibody. With this molecule in hand, we went forward with our first protein transfer experiments to try to induce trogocytosis from one cell to another. To do this, we use an acceptor cell and a donor cell. Our acceptor cells are shown here. It's this Dowdy GFP cell line. It's a B-cell lymphoma harboring GFP in the cytosol so that we can fluorescently track who's an acceptor cell and who's a donor cell. But importantly, there's CD22 positive and CD30 negative. So the cell surface antigen expression profile is really the critical thing to keep track of here. Donor cells that we use, on the other hand, these Carpus 299 cells, are a T-cell lymphoma line. They're CD22 negative, CD30 positive. So the opposite cell surface antigen expression profile to the Dowdy acceptor cells. Now we can do an experiment where we have donor cell and acceptor cell together in a co-culture with the trogotac molecule or parent antibody unconjugated control. And if we're successful, we should be able to see CD30 transfer from donor cell to acceptor cell And this is something we can read out using flow cytometry, using a CD30 detecting antibody that specifically binds to a domain on CD30 the Trogotac does not bind to. Okay, so schematically what this looks like is shown here. Donor cell on the right, acceptor cell on the left, Trogotac in the middle that induces CD30 transfer from the donor cell to the acceptor cell. By flow cytometry, what this looks like with no treatment, just the two cell types, is shown here. We have two different populations. The Dowdy GFP cells are in the top left. They're high on the y-axis because they're positive for GFP, but they're far to the left because they're negative for CD30. Now, those Carpus 299 donor cells on the bottom right have the opposite profile. They're down on the bottom, low for negative for GFP, but they're far to the right because they're high in CD30. Now, with treatment, we can treat with Trogotac, and now we see at the far right this dramatic shifting from left to right of the acceptor cell population. And that's not what we see with the antibody control. And so by flow cytometry, we can see that shifting from left to right, indicative of CD30 acquisition by the acceptor cells. We can take a closer look at this, though, using a technique called imaging flow cytometry. And now these green cells, these are our acceptor cells, the Dowdy cells. And the red punctate labeling on the surface, that's CD30 being transferred from donor cell to acceptor cell. This punctate localization that starts to spread out in some cases, that's indicative of trogocytosis. But at this point, we wondered, okay, so we have this designed system to induce trogocytosis, presumably through the action of CD22. But to verify whether this was indeed a CD22 receptor-dependent trogocytosis event, we knocked out CD22 in our acceptor cells. And what this does is it completely ablates protein transfer from donor to acceptor cell. Another way that we looked at receptor dependency of the process is by blocking CD22 using a ligand antibody that specifically binds to and blocks the ligand binding site on CD22. So there's a non-genetic approach to do the same thing, and this also completely ablates protein transfer from donor to acceptor. But all of the data that I've shown so far is actually consistent with an alternative mechanism. You can imagine a different scenario in which donor cells release small particles containing CD30, such as extracellular vesicles or shed proteins. Then the Trogotac might be able to capture and transfer CD30 from these particles to the acceptor cells. To verify whether this was indeed taking place or whether it was a contact-dependent mechanism, we physically separated donor cell from acceptor cell, but using a microporous membrane that allows free passage of shed proteins, extracellular vesicles, and other small particles. What this looks like, if you reduce this idea to practice, is a transwell setup, where we have donor cells and acceptor cells sharing the same media, but they're separated by this microporous membrane between donor and acceptor. And under these conditions, we also completely block protein transfer from donor to acceptor, indicating that it is a contact-dependent process, and it is trogocytosis that's taking place. I'm not going to go into too much detail around the optimization efforts that we took to establish this Trogotac platform, but I do want to give you some insight into what parameters were most important during this work. So we looked at ligand identity, we looked at different synthetic sialicides, as well as the natural ligand for CD22 and control disaccharide glycans. The takeaway from this was that high affinity synthetic sialicides, like the one highlighted here, were key to driving efficient protein transfer. We also looked at ligand to antibody ratio, so we can vary the amount of ligand decoration on the antibody. And while many different ratios can work well, we find that you get an optimum once you hit 6 or higher. And so sufficient amount of ligand for higher avidity effects are required. And on the other side, the CD30 binding arm, antibody affinity matters as well, but as long as you get into the single-digit nanomolar regime, every Trogotac molecule works about the same. With this kind of optimized molecule in hand, the next thing we looked at was the concentration dependence and the potency of protein transfer. And these molecules are actually incredibly potent for protein transfer, with an EC50 of transfer in the subnanomolar range, about 200 picomolar for an optimized molecule. You see a normal dose response curve as you increase concentration in this low nanomolar range, but as soon as you get into the sort of higher nanomolar, double-digit nanomolar range, you start to incur this hook effect. This is an effect that's well known and well documented in the induced proximity literature. In this case, we think that a similar sort of phenomenon is happening to a ternary complex formation. It's just that the two proteins we're interacting with happen to be on different cells. And so you can saturate at high concentrations CD22 and CD30 binding sites on acceptor and donor cell without inducing cell-cell contact, which is sort of like the analog to ternary complex formation. So now we had this concentration and molecule in hand, and we were able to establish all of that using proof of concept cell line studies. It allowed us to do this optimization and look at receptor and contact dependency of the mechanism. But our next question was, okay, we can do this in cell lines, but what about primary human B cells? Can we translate this to a more physiologically relevant context? To do this, we isolated healthy donor PBMCs from whole blood. We further isolated primary B cells and labeled them with cell trace violet. Now, cell trace violet, this dye, takes the place of GFP in our ability to track who's a donor cell and who's an acceptor cell. Co-culturing our donor cell and primary B cell with a Trogotac molecule leads to a similar profile as we saw with the cell lines. You only see significant shifting of the acceptor cells from left to right with the Trogotac treatment at the far right. And you can look at this again by microscopy, and you see punctate localization of CD30 on the surface of acceptor cells using the Trogotac molecule, but not with the control treatment. You may have noticed that there's a new population that we see on the previous slide. This new distinct population of cells in the top right. These double positive cells are actually intact conjugates between Carpus 299 cells and B cells. This is all we get to sort of learn from typical flow cytometry, but using imaging flow cytometry, again, we can take a closer look at this. And we find evidence for these intact conjugates that are still present at the end of an experiment. And if you look closely, you see that in many of these events, there's a focusing effect of the red signal for CD30 into the synapse between Carpus 299 cell, donor cell, and acceptor cell, the B cell. So we think that this focusing effect is important for thinking about how you can drive selectivity and protein transfer from one cell to another. So far what I've been talking about in terms of two cells coming together is this phenomenon that can take place called trogocytosis. A B cell comes into contact with a target cell, and trogocytosis can occur if we have an appropriately designed Trogotac. But cells come into contact for many other reasons in a normal biological system. CD8 T cells come into contact with lymphoma cells or virally infected cells before eliminating them. And macrophages come into contact with apoptotic cells before phagocytosing them. These are just some examples, but the list goes on and on. So when does trogocytosis happen? When does contact lead to trogocytosis? Is it only certain cell types that can do this? Maybe it's only certain receptors that are efficient in triggering trogocytosis. Or perhaps there are biophysical constraints that limit when trogocytosis will or will not occur. We started looking at some of these questions, and I'm going to give you a sort of small vignette around a number of these questions over the next few minutes. One of them is this question of, can all cell types do trogocytosis? To look at this, we took HeLa cells, which have no business expressing CD22. They're not B cells. It's an epithelial origin cancer cell line. But expressing CD22 engenders HeLa cells with the ability to do trogocytosis triggered by a Trogotac. We find that the wild-type HeLa cells don't undergo trogocytosis, but those stably expressing CD22 do. And so that broader sort of context of the B cell surface and machinery intrinsic to a B cell is not required for this event to take place. What about other targets, other receptors that can potentially participate in trogocytosis through trogotac targeting? We looked at other molecules beyond CD30, including the alpha subunit of the IL-2 receptor that we can transfer from a target cell to a B cell. We can target the immune checkpoint molecule, PD-1, removing it from a T cell and transferring it to a B cell. The same can be carried out with EGFR, receptor tyrosine kinase, on the surface of epithelial origin cancer cells. And the same can be said with HER2, another receptor we can directly transfer from target cell to acceptor B cell. And so that sort of covers some of what we've looked at around the antibodies that can target different receptors and expand the scope of trogocytosis to different receptors. But what about on the acceptor cell side? Is CD22 the only receptor that can be used, and therefore B cells the only cell types we can transfer proteins to? The answer is no. I won't show any data around this today, but we have other receptors as well that can be used to target protein transfer from one cell to another in a fairly modular sense. Early on, I mentioned that trogocytosis is this process by which proteins, but also the plasma membrane fragments they're contained within, can be transferred from one cell to another. And so, with that being said, you may wonder whether CD30 is the only thing that's being transferred, or what about other components of the plasma membrane? Can other proteins be transferred? Can they go along for the ride in sort of a bystander protein transfer process? The answer is yes. We looked at a number of different proteins expressed at high levels on donor cells, but not on acceptor cells. And we find that while CD30 transfers to the highest level, many other proteins transfer as well. What about the reverse direction? Can CD22 be transferred back to Carpus 299 cells? And what about other receptors, not just CD22? Can the bystander process take place in the reverse direction? The answer is yes, but to a much lower degree. So B cell surface molecules can transfer back to the donor cell, but not with nearly the efficiency that you see in the forward direction. The last sort of aspect of this question broadly of when does trogocytosis occur and when does it not occur revolves around biophysical constraints. And I think the best way to introduce this question is to make an analogy to a better studied system, which is when a macrophage comes into contact with an apoptotic cell. If we zoom in on the synapse between these two cells, the synapse distance, the distance between the surface of the macrophage and the surface of the apoptotic cell, it's controlled by the site at which an antibody opsonizing the apoptotic cell binds. It can bind close to the membrane of the apoptotic cell, inducing a short synapse distance, or you can imagine an antibody recognizing an epitope far from the surface of the apoptotic cell, leading to a wider synapse distance. And this actually has a profound effect on the efficiency with which the macrophage will undergo phagocytosis and eat that cell. The narrower synapse gives rise to higher efficiency of phagocytosis. And so we wondered, okay, what about for trogocytosis? Is there a rule here as well where synapse distance controls the efficiency of trogocytosis, this other fundamental immune function? We can't, unfortunately, test this using our first generation molecules because the CD22 ligand we have binds to domain 1, the most membrane distal domain, and no other small molecules exist that bind to any other domain on CD22. Instead, we transition to a second generation platform where we have a nanobody recognizing CD22, where now we have the modularity to generate nanobody fusions that recognize different domains of CD22. Domain 1, the most membrane distal, domain 4, about halfway through, domain 6, close to the membrane, and domain 7, the most membrane proximal. Now, with this systematic variation in the intrasynaptic distance, we can look at the effect on protein transfer. And we do see a significant effect, the opposite effect of what you see for macrophage phagocytosis. The widest synapse gives rise to the highest level of protein transfer, with the most narrow synapse giving rise to the lowest levels. So this is the opposite of what you see for macrophage phagocytosis. It's also the opposite of what you see for T cell killing. And while we've only studied this in one context so far, if this is a general trend, this better equips us to engineer antibodies to avoid or promote trogocytosis, depending on the context. So all of that kind of revolves around this question of when does trogocytosis occur and different aspects of it. But the last story I'm going to share with you today centers around a second question, which is what can trogocytosis proteins do? Once a protein is transferred from one cell to another, is it still functional? Can it imbue the new cell with a different function? And the first place that we looked at this is in addressing antigen escape in tumors. This is a process by which, under therapeutic pressure, an antigen positive cancer can undergo depletion of antigen positive cells, leaving behind only a small number of low or negative expression cancer cells. They can then grow out, and you can end up with an antigen escaped cancer. This is a common resistance mechanism, and one where we wondered whether we might be able to use targeted protein transfer to intervene and resensitize cells to a targeted therapy, such as a T cell engager or a CAR T cell, ADC. To first sort of do experiments to address this question, the first thing we needed to do was look at the function of a targeted therapy, in this case a T cell engager, in a new cell that doesn't actually have the ability to express that target. And so we looked at MHC class 1 to do this, transferring it from one cell to another, using a second generation Trogotac molecule that can induce bystander transfer, so we don't have the issue of the antibody blocking function of the transferred protein after it's been transferred. We can do this transfer with fairly high efficiency, but the critical question is not whether we can transfer the protein at this point, but rather whether this protein, now adoptively expressed on a target cell that doesn't actually have the genetic material to express it in the first place, whether it can be used to redirect T cell killing function. And the answer is yes, we can use a T cell engager to recognize that transferred protein and induce T cell killing of the target cell. This has important implications for our thoughts around what we might be able to do with rewiring tumor microenvironment antigen expression and changing therapeutic targeting, but it also has important implications for T cell engagers that are currently in the clinic around the world. So going forward, there's a lot of unknown sort of mechanistic questions around trogocytosis in a fundamental sense. I think if we can answer those in more detail, this will give rise to therapeutic applications of this space and hopefully eventually close the loop in this area. Just to wrap up, none of this would have been possible without my postdoctoral advisor, Carolyn Bertozzi, as well as our collaborators, lab and admin staff, and this work was funded by HHMI and the NIH.